You're at the cutting edge of cardiovascular sciences, and I'm your host, John Cook. I'm professor and chair here of the Department of Cardiovascular Sciences at Houston Methodist Hospital. And joining me in the studio today is Dr. Lee Lai, who is an assistant professor in the Department of Cardiovascular Sciences. And Dr. Lee Lai is going to be introducing our guest today.
Good evening, everyone. So today it's my great pleasure to introduce Dr. Ron Wang, who is our speaker today. So Dr. Ron Wang is a professor and a Mildred V. Strauss Endowed Chair and Director, Laboratory for Accelerated Vascular Research in the Department of Surgery at the University of California, San Francisco. Dr. Wang got her PhD at the University of North Carolina, Chapel Hill, where she pioneered the study of angiogenesis using cystic embryonic bodies. Actually, our lab is used to use this method to differentiate human iPSC to endothelial cells. It's good to know who is developed this method. And Dr. Wang did her postdoc fellowship in the laboratory of Nobel laureate J. Michael Bishop at UCSF, where she joined the faculty there and focused on the molecular programming in arterial venous specification, concentrating on the Notch and the FAB2 pathway underlying those processes. Her lab also explored implication of these molecular pathways in vascular diseases such as stroke, using sophisticated mouse genetics with cutting-edge 2-photon live imaging capabilities. Welcome to the show, Dr. Wang.
Thank you, Lily. Mm-hmm. I really appreciate this kind invitation from Dr. Cook and you, and I'm really happy to participate in this webcast.
Well, we're really interested to hear what you have to say, Dr. Wang. You've been doing some amazing work in arteriovenous malformation, understanding the genetics of it, understanding how this abnormal vascular aberration occurs. And it's really a significant clinical problem, which can cause stroke. It can cause disfigurement if it's affecting the face. So it's really a difficult problem to deal with also surgically. So maybe we can talk about your work in that context during the discussion. But I think first you're going to be telling us a little bit about the science behind AVMs, correct?
Absolutely. Yeah. We entered this field serendipitously. And we were just studying arteriovenous programming, and our mutant mice developed AVM, and that's, to my knowledge, one of the most robust, 100% AVM with all hallmarks of AVM. That's opened this area of investigation for us.
Tell us
we published the first paper 2005. At that time, not much. Certainly not much as known as now for AVM, and not as many people in the broader angiogenesis field were paying attention on AVM.
You said
and now we see the whole transformation of the field. It's really gratifying.
You said that it was serendipitous that you got into this field. Tell us how that happened. So that was back in 2005, you said, that you started getting into arteriovenous malformation, the science of that.
My journey started much earlier. So when I started my lab, we were fascinated by the developmental biology field and showing that there is a distinctive arteriovenous molecular programming, which I'll touch upon later. At that time, before that, I was already interested in Notch4 because it's an oncogene. When it's discovered, it's oncogene, but when it's cloned, the homolog of that oncogene is Notch4. Well, oncogene is called IN3, integration 3. And because I was in Dr. J. Michael Bishop's lab, and he won Nobel Prize because of the discovery of oncogene. And so IN3 is an oncogene. The homolog is Notch4, which expressed in the vasculature very specifically. That's how I got interested in. And furthermore, joining the developmental biology study and understanding the arteriovenous molecular programming and Notch signaling also play a major role. In that context, we made mutant mice expressing Notch4, which I'll talk a little bit more later. And turned out that those mice develop AVM. At that time, I must say, we didn't predict they would get AVM. We did not know. And then with all the meticulous characterization, those mice indeed exhibit AVM phenotype 100%. And this has been followed up. This type of research has been followed up by many investigators, many labs since.
You're in a department of vascular surgery at UCSF. Tell us how you work with the vascular surgeons, how you work with the clinicians, what you've learned from the clinicians that maybe has informed your work. Tell us a little bit about that kind of interaction between the clinicians and the scientists.
That's a great question. It started with even when I was looking for a faculty position from a postdoc, and that's before the time before translational research become more known. At that time, I just have a single goal that with all my basic science education and training and the skill and expertise, and I wanted to contribute to something in my future career or in my faculty position, contribute to something understanding disease and finding treatment for disease. I felt that human health and diseases are the biggest challenge of our time. And DNA double helix was discovered 1953. And at the time I was looking for faculty position 50 years later, we still know many genes, but we still have so few cure and so few drug. So even today, I feel that is the greatest opportunity of our time. And I often tell my students, we now have great car, great computer, and great phone, but we still don't know as we would like to know about the disease and particularly how we treat them. So, so basically, I choose to join clinical department, and in that context, I have been continuing learning, and as if I'm going to our mini medical school and interacting with clinicians, vascular surgeons on a routine basis. Both me and my lab has been in that environment ever since. So I hope I answer your question that we certainly, when we have questions about our research related to clinical aspects, not only the vascular surgery division, our department, and UCSF as a whole, my lab is very well connected to the clinic, to the research related to disease at UCSF and beyond.
I liked what you said about science in the service of humanity. That's struck a chord. We have folks listening in, and I'm going to give them an opportunity to ask questions during your talk. So if you don't mind, I'm just going to let them know how we do that. So for those of you who are listening online, you can text DeBakey to 37607. So once again, you dial 37607 and text DeBakey, and then you can just text in your question to Dr. Wong. If you're online on your computer, you can go to pollev.com, enter DeBakey, and then respond to that activity. Do you have any questions for Dr. Wong before we start?
I think I would like to hear her talk first.
Yeah. Why don't we go to your talk, Dr. Wong, and then we'll have a little discussion after that.
Sure. So the title is molecular. My talk title, as I posted, is molecular regulation in arteriovenous programming and arteriovenous malformation, or AVM. Today, the molecular regulation is going to be centered on Notch signaling in this process. So, how can I not advance? Oh, here we go. So Notch is a very interesting gene, and I wanted to bring this up, how basic science shape our understanding of very important medical related conditions. So Notch was identified, this mutant, Drosophila mutant with a Notch wing blade, was identified in the 30s in famous Morgan Drosophila mutant screen. And through many efforts, Notch gene is known in Drosophila to decide the cell fate of two progenitor cell, or in our population of a progenitor cell, they have two potential fates. And Notch determining one fate, in fact, without Notch also determining the other fate. So Notch is known to be universal arbiter of cell fate decision. So Notch determining bipotential cell fate. That is the classical function. The reason the mutant get Notch wing blade is there's a loss of epidermal cells. And the reason Notch mutant exhibits this is there is a population of cell can give rise to epidermal cell or neuronal cell. And those expressing Notch become epidermal cell, and those without Notch become neuronal cell. If you have a Notch mutation, you have a loss of epidermal cell, therefore Notch wing blades. It's a beautiful study, and later the Notch gene is cloned. What's really lucky and interesting is in the vascular system, you can see this expression pattern and showing not one of Notch ligand, but Notch receptor, and other Notch ligand and Notch mediators are exhibiting, or Notch downstream targets are exhibiting similar pattern that is expressed in artery, not in the vein. And in fact, many studies have through zebrafish mouse have shown that Notch determining arterial and venous fate. Super interesting. And this fate decision may occur before the first embryonic heartbeat, which challenge or advanced the understanding of what determines artery and vein. In textbook right now, perhaps it's still written that the differences between artery and vein are determined by the hemodynamics, largely. And here there is this genetic programming determining artery and vein are certainly provocative and open up a new study ever since. So it's a very exciting area of study. I must also say that those genetic programming may actually mediate hemodynamic signaling. Even we think it's making sense that many of the differences of artery and vein are because the hemodynamic differences exposed to the different vessel segments. However, we don't know well how those hemodynamic differences are converted into biochemical cues that mediate the biological and the pathological differences between artery and vein. So I just want to connect the genetic programming with the known hemodynamic signaling at the molecular juncture. So let's take a look at NACH. It differs in Drosophila, in zebrafish, or other model organism, and in mammalian system. And this is a slide showing NACH signaling in mammalian system, mouse and human. There are 4 NACH receptors in mammal, and 1 through 4. They are type 1 transmembrane proteins. And NACH1 and 4 are expressed in endothelial cell. NACH3, for example, is uniquely expressed in smooth muscle cell. NACH2 are expressed more, not as uniquely. And NACH4 is expressed primarily in endothelial cell, while NACH1 actually expressed in many other cell types. And that's why my lab has been focused on NACH4 for a long time. And the NACH receptor is activated when it's bind to the ligand, which is also type 1 transmembrane protein sitting on the adjacent cells. So it does mediate cell-cell communication, cell-cell signaling. And the ligand, type 1 transmembrane protein, there are 5 of them in mammal: jagged 1, 2, delta 1, 3, and 4. And the one expressed in endothelial cell are highlighted in here, and many of them expressed in smooth muscle cell as well. And my lab is more focused on endothelial cell. My talk is a little bit endothelial cell centric, so those expressed in endothelial cell are highlighted. So the activation of NACH receptor through ligand binding and this cell-cell contact influence NACH receptor get activated. So what it is, this activation, following this activation is cleavage of NACH intracellular domain by gamma secretase. And this intracellular domain, so-called NACH ICD, then get translocated into nucleus. And there it kick out transcription repressors and then contact the transcription factor and leads to NACH downstream target transcription. That is how NACH from cell-cell communication to the downstream transcription, mediating the cell fate decision. I'm coming back to NACH4, and here I call it NACH4 star, but this is the mutant originally named as the integration 3. And this IN3 is cloned in Harold Varmus's lab, or identified in Harold Varmus's lab. And by the way, Dr. Varmus co-won Nobel Prize with Dr. J. Michael Bishop. The 2 labs were together, running together, and at those exciting time. And so I did my postdoc in Dr. Bishop's lab, and also very, very interactive with the work and colleagues from Harold Varmus's lab. And their lab and the subsequent colleagues. And the IN3 here is also called NACH4 for simplicity. Is a mutant that without extracellular domain, and this mutant is constitutively cleaved, so constitutively active, the ICD, NACH4 ICD, will be translocated into nucleus and mediating NACH signaling. And I will be talking more about this mutant in the subsequent talk. Okay. Any questions so far?
Keep, no, not at the moment. Please continue.
Okay. And then through this paradigm, it's really interesting, and many people's work I listed in here, and some may have not been included. But there is a whole field of awesome developmental biology, and really reveal that there is, at least in endothelial cell, are signaling pathway determining arterial molecular fate versus venous molecular fate. So, for example, Notch ligand delta-like 4 cannot activate the Notch receptor, and leading to downstream target FMB2 expression and repressing EPHB4 venous marker expression, leaving a molecular arterial programming. In the vein, then, CoopTF2 sit upstream of Notch, repressing Notch, also repressing FMB2 expression, and actually allow EPHB4 venous marker expression and providing a venous molecular programming. But this is quite well known, but what we don't know is exactly the physiological function and the pathological ramification of this important genetic signaling or in development, and we wanted to know more about the postnatal function of this pathway. And that's where leading me to the AVM. I should have given a little bit better introduction, but basically my lab made, or me, at the juncture of, towards the end of my postdoctoral fellowship, I made a mouse model where Notch is expressed in endothelial cell through our tetracycline inducible system. Basically, we express Notch in endothelial cell. We can turn it on and off, and it's only in endothelial cell, very specific. And that mouse, it's published since 2005, developed AVM. And so let me give a little bit of introduction of AVM. This is a disease Dr. Cook was talking about, disfiguring in many cases. Very dangerous when it's happening in vital organs such as the brain and lung and many other tissue. So as you can tell, this is a very difficult disease to treat. No molecular or no drug, and there are interventions such as surgery to remove it, but it's very, very dangerous. And there was a Aruba trial. Basically, is asking whether the natural course versus surgical intervention, which one is more safe, or whether surgical intervention is beneficial in treating the disease. It's a very controversial trial, and it's very I wanted to say it's complicated, but the trial was ended because that the surgery, as collected, the data collected were more, it were not beneficial for patients. So as you can, I don't want to get into detail of that trial. It's very controversial, but we through this study, we understand how difficult to treat this disease, and there's no safe, and drug. So AVM from its core pathology, and this is a cardiovascular audience, so I don't have to introduce in great detail, but we all know that artery, the fundamental microcirculation is artery deliver blood to arterial and then perfuse the capillary and nutrient and waste exchange, and the waste blood return from venule to vein. That's a normal microcirculation. However, in AVM, it's clear artery directly shunt blood to the vein, displacing capillary, so there's no nutrient and oxygen exchange, so you suffer ischemia. At the same time, this high flow shunt, AV shunt, is also prone to hemorrhage. So therefore, patient also suffer from a hemorrhagic insult. So this is a disease deadly or detrimental for two major problems. And that's basically the core pathology. How it occurs is completely unknown. We basically propose a hypothesis that aberrant arteriovenous programming may be a molecular mechanism of AVM formation. We are still continue pursuing this, and many other labs are sharing their results in this line of study. So this is the evidence leading us to for that hypothesis and a proposal. So this is another molecular marker, arterial marker, FMB2, showing in blue and not expressed in the vein. It's only expressed artery, not in the vein. In arterial, yes, but not in the vein. However, in the Notch mutant mice, we developed in here, we call Notch 4 star mice. Again, those mice express Notch 4 in all endothelial cell and in an inducible manner. And those mice develop AVM evident by those tortuous vessel connecting artery to vein directly. You don't see capillary in this image in the mutant. And what's interesting is the vein, which is, I hope you can see my cursor, right? You can see, can you? So basically vein is, unlike the control vein, express FMB2, as well as the AV connection or the AVM segment, they also express FMB2. So we show in this mice that we reprogrammed molecularly, at least, the vein and the AV connection. So unlike in the control, there is a specific demarcation of artery and the vein molecularly. And by this molecular identification, that vein also expresses arterial marker as well as the AV connection. And the venous marker, similarly, in the control, this is the EPHB4 venous marker, and it's expressed in the vein, not in the artery. But in the Notch mutant, if you look at the middle panel, the venous marker are missing, consistent with the Notch arterialized vein and repressed venous molecular programming. What's interesting is, as I mentioned, this transgene, we can turn it on and off. Even in the affected AVM mice, in AVM, we turn it off, we can actually lead to the reversion of arteriovenous programming, as well as AVM, as well as clinical recovery of the mice. I'll show you the data in a little bit. But this data basically suggests that Notch fully capable and indeed arterialized the vein in this mice and in a reversible manner. So this is a summary of the data I just showed you. We propose in the control that there is arterial molecular identity as well as venous molecular identity. In the Notch mutant, when Notch is on, the venous molecular identity were repressed, and the Notch reprogrammed the vein and leaving arterial molecular programming in the mutant vein. However, this is reversible. We can turn off Notch and restore the venous programming. And using this mouse model, at the same time, we also develop our collaborating with bioengineer laser physicist. We built a 2-photon microscope that's not commercially available, specifically benefits in vascular imaging. It's a so-called 5D imaging. We built a cranial window, a mouse skull, and the mice will be fixed on steel tacks, and it can go into imaging the precise spot over days. So we can, for the first time, uncover the disease initiation, progression, and regression in a 3D at single cell resolution over time, 4D, and with measuring the velocity of the blood. So this 5D imaging is an amazing approach, and for us to learn many things. Number one, we were for the first time to find that the AVM, the large vessel, originates from capillary. So this is random imaging, and then we go back once we find the AVM, trace back, and they all come from microvessel, and as shown here. We also actually, let me advance. We also were able to show that the AVM can also reverse back into microvessel, and under this 5D imaging. So for example, in this case, we found AVM with abnormal vessel topology as well as the hemodynamics. You can see they have a high flow and very turbulence. And as we turn off Notch and this turn Notch 4 off, and the abnormal vessel and the hemodynamics quickly reverse to normal. They did not disappear. It's a safe return. And this paper published in Science Translation Medicine featured on the cover, and our perspective, basically really highlighting the importance. And clinically, we only can use surgery and intervention to remove AVM. This is the first time we can safely turning off just one gene, leading to safe regression of AVM, returning our dangerous AVM with high flow to our low flow microvessel. Amazing. And then I also wanted to mention that not only AVM pathology can be normalized. Mice suffer, those mice are 100% suffer from hemorrhagic stroke, or they display neural dysfunction, just like many humans suffer from neuronal deficit. And the mice, once we turn off Notch for the same mouse, quickly recover ever since. So I'm going to go back to talk about that this quick formation over days and the safe regression, and leading us to think, what did Notch do to the mice, to the vessel, leading to AVM? There are obviously many hypotheses. one of the one we pursued for a few years, and hopefully we'll publish soon, is a paper in press right now. Paper in revision, minor revision, not in press yet. I hope it's in press. And we are pursuing this hypothesis that Notch actually affect the vascular tone in those mice, and that leads to the AVM initiation. We have many evidence in this manuscript, but this is one of the vivid data that I wanted to share. So this is cannulating the vessel isolated from cannulating artery from the mouse brain, and with the increase of pressure, it's known that the vessel will constrict compared to the maximal dilated stage, which is dotted line on the left. And with increasing of the pressure, the vessel will constrict, and you can see the solid black line. However, in the mutant mice, increase the pressure is unable to constrict the vessel. The vessel failed to constrict and remain maximally dilated. So this data, we have many other data in vivo vascular tone analysis showing that those mice for sure have defects in vascular tone. They are unable to constrict. They are dilated, and that is what we think that AVM is forming. And we also provided evidence, I'm not showing here due to the interest of the time, that ENAS is involved in this process. So basically what we are proposing is Notch4, at least through in part ENAS and NAS signaling, and affect the or compromising the tone of the vessel, and because the lack of the constriction and the flow perpetuate AVM formation.
Dr.
So
Wong,
that's
can I ask you a question about that?
Yeah.
There could be, what you're talking about is the loss of cerebral autoregulation, which is really an important physiological response to changes in pressure in the cerebral vasculature. It's a way to control to reduce the pressure that's transmitted to the brain, to the microvasculature. And if that's lost in the cerebral arterioles, that can cause a lot of problems downstream. Do you know, is this purely a functional abnormality, or is it also a structural abnormality? For example, is the vascular smooth muscle of the arterioles intact in these vessels, Notch4 mutant vessels, that have lost the ability to autoregulate?
Yeah, it's under investigation. Very good question. Mm-hmm. And I don't have the data to show yet, and we have our work in progress.
Okay, all right.
Yeah.
Thank you.
But in this study, we did show that hydrogen peroxide, we use very sophisticated mass spec analysis, collaborating with redox biochemists, and we show that the NAS sensitive hydrogen peroxide production is increased. That could, beyond other factors, and that could also compromise the vascular tone. So there might be multiple avenue leading to the reduced vascular tone, and this is one of the one we nailed down, and with very clear biochemical evidence. I didn't show those data. Hopefully the paper will be coming out soon. I'm happy to share that. So So basically, what I wanted to end is also very exciting. Not only those AVM can safely regress and completely regress the AVM when notch 4 turned on later. And at least 1 month later, we analyzed very little relapse. So there is also hope. What we think is even if AVM were formed, they are able to completely regress. And at least using this model. And the completely regressed organ, and even later, maybe due to age, and when notch 4 is turned on, it will have much delayed, if not, even more prevention of future AVM formation. What I'm trying to say is the regression followed by, even if the same insult is imposed, the body, possibly due to age, I believe is due to age, after treatment, AVM often occur in young people. After treatment in adults, and there might be hope for minimizing relapse. So this is the paper we published in Journal of Experimental Medicine a few months ago. And with that, I'd like to thank the lab member, past and present, for this work, Lawrence Corinne, as well as, well, their name are not here, Patrick Murphy and Tyson Kim, as well as Tim Carson, who initiated this work. And I also wanted to thank many of the current lab members, Xuetao, Cynthia, as well as Hazel, Sasha, for their current contribution. I do wanted to mention the vascular tone data. I want to particularly mention Feng Chen for carrying that work to this stage. I wanted to thank the collaborator, Chris Schaefer from Cornell, the bioengineer laser physicist, whose lab we have collaborated for almost 15 years on this remarkable journey. And Manuel Navido's lab from UC Davis, particularly Matt, then in his lab. And currently Matt is also an independent investigator in Louisville, Kentucky. Their work, what I showed in the isolated cannulated vessel, and Jessica Zenonica from Medical College of Wisconsin is the redox biochemist, awesome biochemist, and that I mentioned in our work, as well as others who contribute to this work. Thanks for the funding, NIH, DOD, AHA, TRDRP, which is California tobacco-related research program. Finally, I just want to give our posting that we are recruiting. If you are interested in this type of work, please contact me. This is my email. Thank you.
Thank you, Dr. Wong. And this certainly does look like a very nice place to do your postdoc, somewhere there in San Francisco. Well, that was fascinating. And I think I'm going to start with a question. But before I do, I just want to let our audience, remind our audience that they can join us with questions. Text DeBakey to 37607. So call 37607, text DeBakey, or you can go to polev.com and enter DeBakey. So thank you for that, Dr. Wong. I have a question for you. I've had several patients with arteriovenous malformations, and particularly when it affects the brain, it's a terrible disease. And as you mentioned, the surgical treatment is not very successful. And another approach has been to use embolic approaches, to feed a catheter into the AVM and use embolic approaches. And that can be very useful if someone is bleeding and is having worse symptoms because of bleeding. But long term, it doesn't seem to cause the AVM to wither. In fact, it seems, I've seen many cases that where you try to treat it surgically or with embolization, and it gets worse. So I'd like your thoughts about that. What is causing it to become worse when you intervene upon it surgically or with a catheter? The other thing I want you to remind us about is embolizing or surgery is not going to be the answer, because you still are left with an area around that AVM or that's perfused by that AVM that is not perfused well. It's ischemic, correct? So tell us about why are these things so aggressive? Why is the AVM so aggressive and resistant to surgical or catheter-based treatment?
Mm-hmm. I'm happy to share my thoughts, and I'm not a clinician treating the patient, and many colleagues may have a better idea. But my thought is that the reason AVM is so aggressive is my thought is because the vessel may have compromised the constriction. So if you imagine your vessel with a high flow without capability or with compromised capability for constriction, and you will get higher flow, keep getting higher flow, and the vessel getting bigger and then bigger flow. So that's our bigger flow, wider diameter, bigger flow. So it's a perpetuating process until disruption, until rupture, or until something terrible happens. Basically rupture. So rupture is the biggest problem. So that is how the destination of the disease by its virtue. It's difficult to treat, and surgically remove, you will have to remove completely. And also, it does not, in my view, treat the roots, which is if it's a vessel constriction problem, surgery is not going to completely minimize that. And furthermore, it's known, I didn't talk about it, our work and others also propose that vessel during repairing active angiogenesis are more prone to AVM formation. So that's why sometimes after surgical resection, and there might be recurrence. Okay. And in terms of Dr. Cook, you asked that sometimes the emboli afterwards is getting even worse, correct? And for that one, my thought is embolization is not complete. You embolize the major conduit or AV shunt, and in fact, that may lead to higher flow in the adjacent vessel, and therefore may push those vessels
to grow,
develop AVM even worse and more of them. So that would be just one thought.
Yeah, that makes sense. Yeah, it's a positive remodeling. Yeah. Dr. Li Lai has a question.
So, Dr. Wang, thank you for your talk. It's very interesting. So I guess I'm more interested in the molecular mechanism of this disease. So in your talk, you show very nicely with all the mice work, you show that notch plays an important role in the AVM formation. But do you see the notch upregulation in the AVM patient?
Absolutely. I didn't show for the interest that I thought I can only allow 15 slides, so I cut many of them. But It's a great question, and it gave me an opportunity to explain that. So Notch and Notch downstream targets, quite a few of them are upregulated in AVM compared to biopsy or autopsy controls. We did that work. We published that work. And then followed subsequently other lab, or about the same time, other lab also reported that, including Notch4 is upregulated. So so far there are quite some publication showing Notch signaling is upregulated in human AVM.
Thank you. And I may have another question regarding the core pathology of AVM, because when you show the illustration of the pathology of AVM, you showed, there is artery vein contribution to the AVM, but there is also capillaries contribution to this AVM, because in the AVM there's it seems like the capillaries are gone. So where do you think those capillaries go, and what do you think happens to capillaries?
Yeah, very good question, and I need to explain this better. When I say core pathology, that's primarily from clinical perspective. So those drawings are from clinical, largely from clinical understanding, i.e. that's when it's well formed, diagnosed AVM are that pathology, i.e. the artery shunt blood directed to the vein, there is less capillary around, so on and so forth. So I talked about that. At the same time, I also talk about in the mouse model that we find that capillary, selected the capillary in Notch to form AVM. So certainly capillary is involved in that. And I didn't say is that as AVM in our mouse model continue, they will develop similar pathology like human. So if you imagine when there's a high flow AV shunt, their adjacent capillaries resistance is too big, and the flow would not go to those capillaries. They will just shunt from the high flow. So those capillaries will have less and less flow. Without flow or lesser flow will leading to their regression and the disappearance. And that continue to feed the AVM formation. And I hope this I connected the dots on this point, and I answer your question, Lee.
Yeah, that made sense. The one question that I have is, why is this process, or how is it that this process is so localized? Because if it is a Notch mutation that's responsible for the AVM, there's something else that must be going on that explains the localization of the AVM to a certain part of the brain or a certain part of the tissue that's affected, like the face.
Great question, Dr. Cook. You're certainly a vascular biologist, so you can ask this question. Insight for vascular biologists. What it is in my view, our view, is when the vessel loses constriction. And so, yes, Notch expression is pervasive, and we also see the capillary enlargement also widespread. And the focal development of this is due to the fact that the vessel don't constrict, and then the flow basically take over. The flow will drive the vessel continuously grow with the most energy efficient. So the proximal and vessel with the topology that can easily adopt this AV shunt high flow will gradually take over the other adjacent vessel. In fact, some AVM will regress. When we watch during the two-photon imaging, it's not initially focal, but eventually become focal. I think this become even more apparent in human case that it may grow even longer time. That's one reason. The second reason is I didn't touch upon is we become more and more realize that in this paper we also include, we only detect 60% of the endothelial cell express Notch4, 40% didn't. So however, the phenotype is strong throughout. So we think that even the chimeric expression or subset of cell express Notch4 star or abnormal can cause AVM. This is related to in human somatic mutation is found in brain AVM. Somatic mutation by definition may be sporadic. So that is another possibility of focal AVM. I hope I answered this, Dr. Cook.
Yeah, that actually makes a lot of sense to me, that a sporadic mutation, a somatic mutation, could be responsible for the localization. That does make sense. And that actually could explain also why sometimes you'll see an AVM in the brain, sometimes it's in the spinal cord, sometimes it's in the face, like the picture you showed us, sometimes it's in the hands. And so when those different tissues are looked at, is the mutation still the same? Is it a Notch4 mutation, or are there tissue-specific mutations that are responsible for AVMs?
Let me just mention, yes, the somatic mutation is a great explanation for focal AVM. But even in our mouse model, Notch4 is widely expressed in vessel segments or vascular bed, and we still see focal AVM. So certainly hemodynamics, what I'm talking about, the flow-driven AVM is also another potential mechanism for focal AVM. As far as the mutation, in fact, the somatic mutation first identified in brain AVM are KRAS mutation, not Notch. And in other AVM, there are hereditary AVM, such as HHD, hemorrhagic telangiectasia, hereditary hemorrhagic telangiectasia, HHT, are certainly a known gene, but in other sporadic AVM, what are the other mutations remain to be identified?
I think we have time for one last question. I'm going to let Dr. Li Lai give it.
Yeah, hello, Wang. So I think my last question is more related to what I'm interested the most, because my studies talk about the cell phase transition from fibroblast cells to endothelial cells under the condition of hypoxia, which is also happening in the AVM. So I'm wondering whether in the AVM you have observed like other kind of cell phase transition, or what's your opinion of the contribution of maybe fibroblast in this disease?
We certainly have not looked at the fibroblast. Our transgene is uniquely expressed in endothelium. So we focus on the endothelium programming and reprogramming. However, we think endothelium sends a signal, Notch in endothelium sends a signal, affect the mural cell nearby. So this is our follow-up study. to look at the adjacent cells, even including immune cells. So it will be interesting to learn more about that. Thank you for the question.
We actually, we just got a bunch of questions from our audience. They just came in, and maybe you can just address a couple of them. One is, can AVM occur? Can it spread to other parts of the body? If you have an AVM localized in the brain, do you see it affecting, popping up somewhere else in the patient?
Certainly, AVM can be multifocal versus, focal, especially hereditary AVM. So whether it's spread, i.e. contagious from one to the other, I don't have evidence to think that. So if the vasculature is abnormal, it can be multifocal.
Okay. And then it's a related question. There was a question about which are the most commonly seen AVMs. If they occur throughout the body, which are those that are more commonly seen?
Well, what's the commonly seen? Well, brain AVM is most dreadful. So I think that draw a lot of medical attention. And dermal AVM, I know many AVM researcher and AVM program are through dermatologists. So especially body cover widely throughout the body. So certainly there's lung AVM, liver AVM, spinal cord AVM.
I guess one last question, and that is, since we talked about the poor outcome with, in many cases, with surgery or with embolization, particularly in the brain, is there something from your work that is coming down the pike that could be useful, a molecular therapy based on your work with Notch4?
We'd love to. We are pursuing those.
Great. Well, that was wonderful, and hope for the future, understanding the cause of AVMs. Thanks to you, Dr. Rong Wang. Thank you for visiting us tonight and joining us at the cutting edge of cardiovascular sciences.
Thank you, Dr. Cook and Dr. Lai, and for this opportunity. And thanks for the audience.
Wonderful. Thank you.
Bye.